Well, thanks, Leo, for stealing some of my punchlines. And thanks for organizing the symposium in honor of Larry, only 35, 37 years too late, but I'm glad we're having it at all to remember Larry. And thanks for the opportunity to speak. So because time is limited, let me give you the take-home message, as Larry would always instruct us at the very beginning, so that in case I run long, you'll have the main thing I want to say. And the main thing I want to say is that becoming Larry's graduate student was the pivotal event of my career. Everything that I did subsequent to leaving Larry's lab was largely a reflection of Larry's training and mentorship, and for that, I will always be grateful. Everything else I have to say today is sort of an amplification of that and more detail. I'll begin by saying a little bit about some of my experiences as Larry's student, and then I have chosen some examples from the work we've done over the years, some of it now quite old, but I think work that best illustrates our approach and how much that was influenced by Larry's training. I became Larry's graduate student almost by accident. I didn't come here for graduate school with the intention of being a Drosophila geneticist, especially not a classically trained Drosophila geneticist. But almost by accident, I ended up doing rotation in Larry's lab, and I immediately fell under his spell. Although I consider myself sort of a naive first-year graduate student, it was clear to me that Larry was a mentor unlike any other I had ever encountered. He could make even the most arcane aspects of chromosome mechanics seem intellectually stimulating and exciting. I don't know anybody else who had that ability. And I decided, it was a very conscious decision, I decided that what I worked on as a graduate student would be less important than the lab in which I received the training. And I decided I couldn't get better training than in Larry Sandler's lab. And so I joined his lab as a graduate student, and I think that was probably one of the best decisions I've ever made. I took these pictures myself. Those were in the days before everybody ran around with a camera all the time in their pocket. But I took these. They're a little bit grainy. But this is how I will always remember Larry. Holding forth in the lab, coffee cup in one hand, cigarette in the other, despite the fact that ether fumes were rampant in the laboratory. Biological safety was somewhat different in those days. Holding forth while we were busy sorting flies, recounting flies, recounting stories about Sturtevant or Muller or Nowitzki, or lecturing us on the importance of using language always with accuracy and precision, or as future professors impressing upon us the importance of professionalism, becoming a good classroom instructor, how to be a valuable member of a department. So even as I'm sitting there, you can't see me, even as I'm sitting there sorting flies, counting flies, I'm absorbing these lessons about being a geneticist and being a professor, about academics. And all those lessons resonated and became very deeply ingrained. This is a different version of Larry. This picture, beautiful picture taken by Kent Golick. Larry underwent this phenotypic transformation after I'd already left the lab. So this is not a Larry that I really recognize, except that I recognize the expression on the face, and I recognize that pointing finger that I was often on the other side of. I have to switch hands. Barry, don't let me ever hear you using data as a singular. Yeah? Some people will remember this. I'm not done. Barry, success in academics means excellence in teaching as well as excellence in research. Barry, if I ever find out you have introduced mites into the laboratory, let me make this perfectly clear. I'm going to autoclave you. I'm pretty sure he wouldn't have, but I wasn't about to find out. But of all the lessons I learned from Larry, by far the most important that Leo has already alluded to, is in Larry's lab, I learned what it meant to be a real geneticist in a way that is, sort of hard to explain if you have not been immersed in it. There's aspects that I can describe. It's not just tools. It's not just techniques. It's not just a way of answering questions. It's a way of making discoveries, of finding the stuff, of discovering things that deserve to be answered. It's an entirely different way of approaching science and a way of thinking about science, and an appreciation of things that are abstract, of phenomenology, and careful phenotypic analysis. It's hard to explain, but I think the best I could say is that if it sounds nebulous, read Muller, read McClintock, and you'll see how real geneticists approach these problems and how they can take a phenotype and make up just so. Geneticists were very successful for many, many decades before we were able to clone genes. And mostly that was based on looking at phenotypes and making up stories, just so stories, that explain the phenotype. This must be jumping genes. This must be telomeres. That's what geneticists did. And you sort of learn how to do that, to think in this abstract way. And that approach to science just became part of me. That is how I think about science. And to summarize it in one way, I would say that to Larry, as he impressed upon us, the genetic approach is always the best approach for tackling any problem in biology that you want to study. And if you absolutely must resort to something else like biochemistry or imaging or some other technique, at least do it in combination with genetics. It will always be better. And if genetics is the best approach to biology, the second half of that is that Drosophila is the best organism for doing genetics. After being out of the lab for 40 years, I've seen no reason whatsoever to change my conviction that those things are true. I would have gladly remained in Larry's lab forever as a graduate student, but Herschel told me he would not give tenure to a graduate student. So I was forced to move on. And in those days, there weren't so many fly labs to choose among, and I really was not sure at all what I wanted to do as a postdoc, and Larry made it very simple for me by making the decision for me. And that conversation went something like this. It won't take long to repeat the entire conversation. Barry, Barry? Seymour Benzer is now working on behavior in Drosophila. But Seymour doesn't really know fly genetics. He needs a real fly geneticist in his lab. He had Jeff Hall, but Jeff Hall has now taken a faculty position at Brandeis. So you should go to Seymour's lab to be a postdoc. I think my view of Larry was somewhat like Leo suggested, but I think also somewhat like the second one as well, because when Larry made that suggestion, I didn't take that as a suggestion or some thinking out loud. I took that as a command from on high. Larry had just told me I should do a postdoc with Seymour Benzer. And so that's where I went to do a postdoc. Even though I knew nothing about behavior and wasn't particularly interested in behavior, and in fact, I found behavior sort of mysterious and hard to get a grip on, mysterious and distant. I didn't know what a unit of behavior was, and so I didn't know how to even think about behavior. But I was very lucky because I arrived in Benzer's lab at exactly the same time as another postdoc, Chenfang Wu, who had trained as a neurobiologist and electrophysiologist in Bill Pack's lab studying phototransduction. And he wasn't quite sure what he was going to do in Seymour's lab either, so we became a team. More than 40 years later, let me say this now in case I forget, Chunfang Wu is my closest scientific colleague, collaborator for many, many years, and a very cherished friend. So I've always been lucky in finding the right people at the right time. And he and I together spent a lot of time thinking about what to do and how to approach the problems we were interested in. And we deliberately decided we were not going to do what was in those days known as behavior genetics, but do neurogenetics, which was a term that was not widely used. In principle, what this means is that we weren't going to use the usual approach that had been done in Seymour's lab, which was to take a complex behavior like learning or courtship or circadian rhythm and try to work your way down to the underlying mechanisms. We were going to turn it around, and we decided we were going to focus on those mechanisms that underlie all behavior. And ultimately, all behavior depends on the ability of single neurons within the nervous system to generate and transmit information in the form of these electrical signals called action potentials. Receive, process, and transmit that information. These action potentials are propagated along axons and relayed between neighboring cells by release of, giving away my punchlines, by release of neurotransmitter at a synapse that activates receptors on the postsynaptic cell, generating synaptic potential, which then triggers another action potential in turn. That's the basics. It's oversimplified, but that's the essence of it. These mechanisms were understood in electrophysiological terms. For example, it's known that an action potential required the activity of proteins in axonal membrane, so-called ion channels, that form voltage-gated pores in the membrane, with an influx of sodium ions inward that drive the rising phase of an action potential, and the efflux of potassium ions that cause the repolarization of an action potential. And as I mentioned, release of chemical neurotransmitters that activate ligand-activated receptors at the presynaptic terminal, postsynaptic terminal. So it was sort of known in electrophysiological terms, but it wasn't really known what this meant in chemical and molecular terms. These are very scarce membrane proteins that had not been purified biochemically. So exactly what it meant for a protein to be voltage gated and so on, the nature of permeation through a channel, those were not understood in molecular terms. This was sort of the dawning of the age of molecular neurobiology as well. So there was a lot of interest in trying to get a handle on these ion channels and related proteins so that they could be better characterized. And we decided our focus was going to be, you could think of this as the neurogenetics of membrane excitability. That's what we were going to focus on. And the question was, how can we get a handle on these proteins? And of course, the answer is genetics. And I think especially for this audience, this slide, it's a generic slide, doesn't need to be elaborated. If you think of the altered phenotype as meiosis, this is everything that, that Larry and his students had worked on. It's the all-purpose genetic approach to unraveling a biological mechanism. It didn't make any difference whether these proteins, the channel proteins, were scarce or abundant. It didn't make any difference if we had any prior information about them. We could isolate mutations in the genes encoding them if we had some idea of what kind of phenotype to anticipate. So doing the electrophysiological analysis as a primary screen was too difficult. We had to choose some behavior that might be indicative of a defect in the signaling mechanism. Not so much because we were interested in studying that behavior per se, but because with the right behavior, that could indicate a defect in the mechanisms we wanted to study. And without belaboring this, I don't have enough time to talk about it, the phenotype we chose was temperature-sensitive paralysis. Flies that were normal at room temperature, but became paralyzed very quickly within a few minutes when raised to an elevated temperature. We weren't the first ones to think about TS paralytics. They were discovered originally in Dave Suzuki's lab at the University of British Columbia in the early 1970s. So here's a graduate student that actually heard Dave Suzuki talk about these mutants, although he was no longer working on them at the time. And at the time we started this work, there were 3 known mutations on the X chromosome. They only looked on the X because, of course, the X is easy to screen. Nobody had dared do this for autosomes. So there were 3 mutations on the X chromosome: para and Shibiri that had been isolated by Suzuki, and comatose that had been isolated in Benzer's lab. No one had touched the autosomes that represent 80% of the fly genome. In addition, another postdoc, previous postdoc in Seymour's lab, had done some very preliminary studies on some of these mutants and had some data that suggested that these mutants might be very good for further electrophysiological characterization. So Chen-Feng and I decided we were going to generate a whole bunch of mutants on the autosomes. I'd never done a large screen in Larry's lab for mutants, but having been Larry's student, I knew exactly what I needed to do, and I knew I could do it better than anybody else in Seymour's lab. And so within a short time, we generated a collection of mutants on the autosomes. At this point, I've lost track. We probably have 50 or 100, most of which we've never had a chance to characterize. They have turned out to be an extremely rich source of material, even better than we had any right ever to anticipate. And as our interests changed over the years, our interests changed, but the game was always the same. It was, okay, now we're interested in ion channels, and we found all kinds of mutants, and I'll talk about some of this work today, affecting ion channels, sodium channels, and these mutants led us to potassium channels. We found regulators of ion channels. Eventually, we went on to study some of this is the work that Leo was a crucial part of in my lab, isolating and characterizing mutants that affected the machinery of synaptic transmission. Then we got interested in the development of the synapse itself. We have mutants affecting synaptic development and architecture. Then we became interested in neurodegeneration, and we found a bunch of mutants affecting. All these are within our collection, originally isolated on the basis of temperature-sensitive paralysis. So the phenotype we focused on changed, but the game was always the same. Yeah, every time we just go back in our collection of mutants, and every time we find interesting mutations that led us to new molecules, new mechanisms, and things that other people had not understood before. Let me just mention, with all these genes that are up here, we've cloned most of these. Beginning in the days when cloning was really hard, and the best tool that we had available in those days was the salivary chromosomes. This was by positional cloning, and the best you could do was to pinpoint the location of the gene on the chromosome and access that region. And in this instance, we had a huge advantage, even over the large labs that were much better molecular biologists than we were, because I really knew cytogenetics. Larry, in his wisdom, had also sent me to Dan Lindsay's lab to learn how to do salivary chromosome analysis, so I became good at that as well. And I also knew how to generate chromosome rearrangements by X-irradiation of chromosomes. So all that arcane chromosome mechanics stuff came in really handy. I knew how to generate the inversions and the deletions and the breakpoints that enabled us to get a handle on the genes. And for most of these genes, that's how we were able to access them. I like this quote, just to give you again, another reflection of my perspective on life. So this is Will Rogers, who was even before my time, an early 20th century humorist and social commentator. He said, I never met a man I didn't like. And I always say that this says more about Will Rogers than it does about the people he met. And that I'm not nearly as nice a man as Will Rogers, because I've met all kinds of people I haven't liked, but I've never met a mutant I didn't like. For me, every mutant is the answer to a biological question, one or more biological questions. It's always the answer. We just have to ask the right questions, and if we're smart enough to be able to listen to what the flies are telling us. So every single mutant is a treasure, and that has been the source of all the important discoveries we've made. So I like all mutants, but I especially like temperature-sensitive paralytic mutants. Again, I'm not going to show you any real data today. I like this because this one idea of looking for temperature-sensitive paralytic mutants has kept me and my students and postdocs busy for over 40 years. This is the first paper I published as postdoc in Seymour's lab on the first mutant we characterized. Oops, I keep hitting the wrong one. I'll talk a little bit more about this work in just a second. Here's the most recent paper we published just last year on one of our mutants in a completely different area, neurodegeneration and mitonuclear interactions, all from the same batch of mutants. So they've been a rich source of material over the years. Let me talk a little bit about the very earliest work we did, because this mutant, NAP, no action potential, it really led us in a fortuitous way to many of the mutants we studied subsequently. So at that time, we had isolated about 6 paralytic mutants. This is the one we decided to focus on. And one of the reasons we focused on this one is it caused very rapid paralysis and very rapid recovery. It's like flipping a switch. The flies become paralyzed as soon as they're exposed to elevated temperature at 37 degrees, literally within 5 seconds, and instantly recover when brought back to room temperature. It's pretty incredible. So we thought, there's not so many ways you can cause flies to get paralyzed that quickly. And we did the electrophysiological characterization, and it turns out that NAP stands for no action potential. It completely blocked the fundamental mechanism of information transfer in the nervous system. It blocked action potentials. For those of you more used to thinking about meiotic mutants, NAP is like the C3G of electrical signaling in the nervous system. It's gone. There's just blocks. Among the many possible interpretations that one might have come up with, based on various evidence, we concluded that the simplest interpretation was that NAP was affecting sodium channels in some way. Of course, we had no direct data. In those days, it was not possible to look directly at sodium currents in Drosophila neurons. And so we had no direct evidence. And there weren't many people who believed that interpretation other than Chenfeng Wu and me. Nonetheless, this is what geneticists do. You tell just so stories. So we said, okay, assuming that we're right, assuming that NAP does affect sodium channels, maybe we can use that information to find other mutants that would also affect sodium channels based on a phenotype very similar to NAP. And by that I mean, not all temperature-sensitive paralytics are alike. They're different in the kinetics of paralysis and recovery. They're readily distinguishable just by looking at their particular form of paralytic behavior. NAP was distinctive. And we went back and looked at the other mutants we had to see whether any of them resembled NAP. And as it turns out, it wasn't one of the mutants that we had isolated, but the first one ever isolated by Dave Suzuki, para, that was most similar to NAP. In fact, behaviorally, they were indistinguishable. Although para is on the X and NAP is on the second chromosome, behavior was exactly alike. Rapid paralysis, rapid recovery. None of the other paralytics had that phenotype. So we did the electrophysiology on para and showed para also blocked action potentials. And then, you give a geneticist 2 mutants with the same phenotype, what are you going to do? We made the double mutant. And the double mutant was unconditionally lethal. And that's the kind of observation that to a geneticist is just rich with information. It immediately told me all kinds of things about these 2 mutants. First of all, I knew that they were affecting mechanisms that were absolutely essential for life. Secondly, it told me that although these mutants were nominally conditional mutants, they were not normal at room temperature. Even though they weren't paralyzed and overtly they looked okay, they were not okay at room temperature. Something was wrong with them because the double mutant was lethal even at permissive temperatures, unconditionally lethal. And third, it told me that there must be some sort of functional overlap between these 2 gene products to account for the synergistic interaction in the double mutant. So our conclusion was that NAP and para have overlapping functions and that they both affect sodium channels. Again, this was not a conclusion that many people accepted. We did manage to get the work published in Nature. And in that paper, we made the proposal, at least a suggestion. We couldn't prove it, but we said we think both of these are affecting sodium channels. And we thought it was important to emphasize this point. This is long before the idea of suppressor and enhancer screens became widespread in Drosophila. The idea that you could gain important information from epistatic interactions in double mutants. To a geneticist, this needs no explanation. It's self-obvious that it would be true, but the nervous system seemed perfect for this because ion channels could be multi-component structures. And even if they weren't interacting physically with one another, you could imagine channels having the same effect on different channels, having the same effect on neuronal excitability or opposing effects on neuronal excitability. And so that you could extract information that would be very difficult to obtain in any other way by looking at double mutants. Not everybody was convinced, including Seymour. And this is an exact quote that Seymour never denied. And I won't say that he was being, completely serious when he said this, but he wasn't completely joking either. I mean, this sort of way of thinking about stuff was not the way that everybody thinks about stuff. And Seymour really wanted more hardcore, real information. This was like one Just So story on top of another Just So story. How can that possibly give you more information? Well, skip ahead 10 years, and the answer is it all turned out to be right. It took us, we weren't working on it constantly over 10 years, but we finally cloned the gene 10 years later, mainly by, this is para, by the heroic effort of one postdoc, Kate Lockney, who single-handedly cloned the gene in the days before PCR with manual sequencing, before we even had good libraries, genomic libraries, or cDNA libraries, with the only information being we knew exactly on the chromosome where para was because we had a breakpoint exactly in the gene. And it turns out, this is how we got into cloning. Para still turns out to be one of the most complex genes in the Drosophila genome. It spans more than 60 kb of genomic DNA. There are at least 26 exons, many of which are alternatively spliced. The open reading frame is 6 kb. The messenger size is 15 kb. We were never able to see the messenger in those days on a northern blot. We had to piece this together from many small cDNA fragments, a lot of which had unspliced introns. I won't bore you with all that. But when we got to sequence, all that effort was worth it. Immediately, it was clear that this encoded protein was the Drosophila counterpart of the rat brain sodium channel that had just recently been cloned by a large Japanese group who had finally succeeded in purifying the protein and cloning the gene from a partial amino acid sequence. So we weren't the first to clone a sodium channel, although in principle, if cloning had been easy, we would have been. I mean, even just cloning based on a phenotype, with all the tools you have today. In any case, it was still one of the first sodium channels to be cloned from any organism, the first invertebrate sodium channel to be cloned. Every invertebrate sodium channel that's been cloned since, as far as I know, has been cloned by homology to para. This includes houseflies, roaches, mosquitoes, many other insects, and that's important because DDT resistance maps to para and all those other insects. And we were the first to characterize in vivo what the phenotype of the sodium channel mutation was. We characterized the first sodium channelopathy. Now, in humans, many sodium channelopathies are known. So we got para, and it was a sodium channel. And the best part of all was it validated, this is now 1989. I'd already been a faculty member for 10 years. It validated everything we had argued up until that point. 2 years later, we got NAP. It's a good thing we started with para because NAP turned out to be a lot more difficult to understand. Ironically, NAP turned out to be allelic to MLE, male lethal, a gene involved in dosage compensation that Mitzi Kuroda and Bruce Baker were studying. And let me take a moment here to remember Bruce Baker as well. Bruce Baker was the senior graduate student, Bruce who passed away, as some of you probably know, just very recently. Bruce was the senior graduate student in the lab when I began, and he became sort of a hero and role model for me. I always wanted to be like Bruce. I aspired to become a graduate student as good as Bruce was, and somehow I never narrowed the gap. After all the years later, whenever I'd see him, I said, I still want to be like Bruce. So it was an honor to have this opportunity to publish a paper with Bruce and Mitzi, and without trying, and this is what's so great about genetics. We clone NAP because of affecting sodium channels and end up cloning the first gene involved in dosage compensation. Isn't that wonderful? Let me make clear that although para is on the X chromosome, NAP is a complicated gene. Its effect on dosage compensation and its effect on sodium channels are entirely distinct. those two functions can be mutationally separated. So the NAP allele itself has no effect on dosage compensation, but it is affecting sodium channels. What that effect was, we didn't know, and it was very difficult to infer because NAP turns out to encode an ATP-dependent double-stranded RNA helicase. And that didn't really give us many insights about sodium channel. That took another 9 years, and another postdoc, Rob, why do I keep doing that? Where am I going? Here, okay. Rob discovered that in addition to all its complex alternative splicing, the paratranscript also undergoes RNA editing. And I don't have time to go through the mechanism of RNA editing, but it involves the formation of a double-stranded RNA hairpin structure, many of which would be appropriate substrates for an RNA helicase. And in the course of looking at editing in the RNA transcript in a NAP background, what Rob discovered was the vast majority of paratranscripts are misspliced by exon skipping events that result in large internal deletions that would result in the production of non-functional sodium channels. In fact, only 20% of the paratranscripts are properly processed in a NAP background. Then we had all the answers to everything we had been working on over that, more than, and now I guess about a 20-year period. Everything made sense. All the interactions between NAP and para made sense, and we could account for everything. I can't go through everything, but we could, at that point, explain everything. So again, it was a wonderful validation that the just so stories we had made up long ago all turned out to be correct. Para was the sodium channel. NAP was involved in regulation of para expression. Ironically, NAP had also led us, based on another double mutant interaction that we were working on in parallel, beginning at the time I was a postdoc in Seymour's lab, led us to potassium channels. How am I doing on time?
I would say maybe about another 5 or 6 minutes.
Okay, that should be enough. Okay. Whereas para and NAP interacted synergistically, we made the discovery that NAP completely suppressed Shaker. That was another mutant being characterized in Seymour's lab at the time by Lilly and Yunung Jan. It had a hyper-excitable phenotype, a phenotype that was consistent with a failure not of depolarization but repolarization, suggesting a defect in potassium channels. And some years later, in fact, Shaker turned out to be, they should separate these buttons a little bit.
Very temperature sensitive.
There, yeah, okay, now, two hands. Okay. Some years later, it would turn out to be the first potassium channel gene cloned from any organism. But at the time, it was just inferred that Shaker was affecting potassium channels based on studies that the Jans had done. So that made sense. We had already suggested that there was an impairment of membrane excitability in NAP, even at permissive temperature. Shaker caused an increase in membrane excitability, and we showed that NAP was able to suppress Shaker at room temperature. Seymour, again, wasn't terribly impressed. It also suppressed the electrophysiological properties of Shaker. So also gave us some information about the regulation of neuronal signaling when you have simultaneously a sodium channel mutant and a potassium channel mutant. But the most important result, the most important discovery, was that in the course of looking at this and characterizing the interaction between NAP and Shaker, we discovered that there were two alleles that the Jans were working on, Shaker 120 and Shaker 133. Shaker 120 they were thought to just be allelic differences in these two mutants, but Shaker had a phenotype that was at least an order of magnitude more extreme than Shaker 133. In fact, they were almost not even quantitatively different, but qualitatively different, which was weird because Shaker 133 was nominally a null mutation. Somehow, nobody had ever accounted for that, and mostly we ignored it. But in the course of studying it, we discovered that when the Shaker 120 chromosome underwent recombination, it lost the extreme phenotype and became more like any other typical Shaker allele. So what's going on there? Well, first we ignored it too, and then I thought harder about it and decided it's trying to tell us something. There must be another mutation on that chromosome that is interacting synergistically with Shaker in a manner analogous to the synergistic interaction between NAP and para. Only this is for potassium channels. So I made up a gene called enhancer of Shaker. And based on the interaction with Shaker, I set out to map this gene that I had made up, enhancer of Shaker, and I found it. So this is the answer. There is a second mutation on the Shaker 120 chromosome that synergistically enhances the phenotype. And I was able to map and isolate that mutation, and it turned out to have a hyperexcitable phenotype, but distinct from the Shaker hyperexcitable phenotype. And when I recombine these two mutations back together, I could reproduce the synergistic interaction. What's going on? It turned out, amazingly, that enhancer of Shaker turned out to be a gene that had been previously discovered back in the 1960s by Bill Kaplan as another ether-sensitive leg-shaking mutation called ether agogo, which in those days was thought to be humorous. So here's this gene that had been around, this mutation, been around for all these years, totally ignored until we rediscovered it based on these double mutant interactions. So EAG has this great phenotype. It interacts synergistically. Why does it interact synergistically? We concluded it's affecting another class of potassium channels distinct from the potassium channels affected by Shaker. It's known that there are multiple different types of potassium channels with different biophysical properties and different pharmacological sensitivities. Shaker was known to affect a specific potassium current. We suggested EAG was affecting a different potassium current. Nobody believed that either. In those days, once Shaker was cloned, everybody who had been interested in potassium channels, and lots of labs that had never had any interest in potassium channels, were cloning potassium channels based on homology to Shaker. And they had the idea that they were going to be able to clone all potassium channels from all organisms based on homology to Shaker. And they said, well, we never got EAG. And I said, yeah, but when you clone by homology, you're going to get all those that are closely related to Shaker. Genetics makes no assumption about anything other than phenotype. And EAG could represent a different type of potassium channel related to Shaker, but sufficiently distant that you're not going to get it by homology. So we cloned it the old-fashioned way, by positional cloning. There was some great cytogenetics that was part of that. I wish I had time to talk about that, but we had to rediscover an inversion back from 1929 that led us to the gene. In any case, this was the work I did with Chunfang Wu, where we described the interaction and the realization and enhancer of Shaker's EAG. A couple years later, it was cloned by Rachel Drysdale, a graduate student in my lab, and, well, a couple years later, this was 83, this is 1991. Further characterized by postdoc Jeff Warmke. And it was very clear once we had the sequence that, exactly as we said, it was obviously related to other potassium channels in the Shaker family, but sufficiently distinct that it represented a different type of potassium channel. In the pore domain and other key domains, it looked like Shaker, but the rest of the protein was very different. It's a highly conserved protein. We find this even in plants. It's been around for a long time. We then went on, we said, well, now we can play the same game. If Shaker is the founder of a family of potassium channels, EAG is likely to be the progenitor, again, of a whole family. Let's find all the EAG relatives. So we played that game and cloned these genes from flies, from rats, mice, humans, and we found, Jeff Warmke did, that EAG was the founder of a family, of which there are three subtypes, ELK and ERG in EAG, represented by multiple subtypes in mammalian genome, one of each in the fly genome. These turn out to be a really important class of potassium channels. This is a review now from 2016, not by us, but by others. On the mammalian EAG family members, they affect neuronal excitability. They affect synaptic transmission. They are associated with phenotypes such as epilepsy, something that we had already anticipated in our publications, important in cognitive function, and so on. They're a very important class of potassium channels, and there's a lot of work. But the best was still to come, and I think I will still finish on time. We mapped the human member, ERG, EAG-related gene, HERG. We mapped it to human chromosome 7. At the time we mapped it to chromosome 7, a group that was studying a familial form of cardiac arrhythmia called long QT syndrome, refers to this interval in the electrocardiogram. They had a familiar form of cardiac arrhythmia that leads to ventricular fibrillation and sudden death. And they mapped it to chromosome 7. But they said there are no interesting candidate genes on chromosome 7. And I said, well, we have potassium channel gene that just mapped there. That would be a perfect candidate for cardiac arrhythmia. So they tested HERG, and it turned out HERG was the gene responsible for long QT syndrome. They promptly got themselves on every newspaper, every TV program saying, we've discovered the first gene involved in cardiac arrhythmia. I think we discovered the gene, and you guys just mapped your phenotype to the gene that we discovered. But anyway, so that turned out to be really interesting. HERG is a gene responsible in humans for cardiac arrhythmia, and the story still gets better. Gail Robertson, who was a postdoc in my lab and then got her own faculty position in physiology at the University of Wisconsin, focused her whole career now on these HERG potassium channels. She demonstrated by expressing these channels in a heterologous system that these channels mediated a potassium current that was recognizable as the primary current responsible for repolarization of the cardiac action potential. The major current responsible for repolarization of the cardiac action potential. And that turned out to be important because in addition to the familial form of cardiac arrhythmia, which is rarer, much more common was a drug-induced form of cardiac arrhythmia, for which many drugs on the market were known to cause this kind of ventricular fibrillation and sudden death. Seldane, Vioxx, many drugs. And it turned out that all those drugs were having these side effects in all the same way by causing blockade of HERG potassium channels. Once that discovery was made, here's a review on that work. Once that discovery was made, the FDA mandated that every single drug, and it's true in Japan and the United Kingdom, as far as I know as well, maybe other countries as well, every single drug that comes to market must be tested for its electrophysiological effects on HERG potassium channels. Every single drug. And so you can imagine the number of drugs that never even make it to market before they ever get close. They must be tested for their effect on this HERG potassium channel. This has generated huge, big business and made lots of people millionaires because their business is to run these tests on HERG potassium channels. That's what they do. We don't get a penny out of this. But this was our discovery, and you know what I like about this is that here's a case that it has completely transformed the pharmaceutical industry, something we never set out to do. It has literally saved, not in some abstract way of translational stuff. This has literally saved thousands of lives by pulling these drugs off the market. My daughter, who has asthma, was taking Seldane at the time. So it's saved thousands of lives. And for me, the best part is this is the ultimate answer. Why do you do genetics? Why do you waste your time doing basic science? Whenever anybody asks me, why do you study basic science in Drosophila, I just say HERG potassium channels. In the most recent check, our papers have been cited. Our papers on HERG potassium channels have been cited 1,000 times. But when I just checked Google Scholar recently, there are now at least 55,000 publications with HERG potassium channel in the title, which is an underestimate because the cardiologists hate the fact that we've made this important discovery that it all has a fly origin. So they've changed the name. It's not HERG, it's KCNH2. It's hard to trace that back to flies. So 55,000 publications on HERG potassium channels, from which I infer that our paper has been cited 54,000 times less than it should have been. I'm going to skip other stories. Suffice it to say that there's a lot of genetic stories like this that I could have told. But let me come to the conclusion. And I'm not going to belabor this. I've made all these points. Just that these mutants have been an enormously rich source of material for studying neurobiology and continue to be. I think these are really the important points, is that most of those important discoveries came from forward genetic screens, the kind that NIH loves to hate, not based on any particular hypothesis, just some good ideas. And where we discovered things that we had never even anticipated we were really after. In almost every instance, the discoveries we made in flies have very important ramifications for human disease. And another lesson, if there's anybody here from NIH, NIH hates this stuff in the modern genomic era. We no longer call it genetics in my lab. We call it functional genomics at single gene, functional genomics at single gene resolution, which means isolating mutations and phenotypic analysis. These approaches, I will argue to the last breath I take, these approaches have never been superseded. Of course, we should take advantage of all kinds of genomic approaches, but that doesn't replace this approach to biology, which I still think is the most powerful method that has ever been found. And let me end where I began, by thanking Larry. I hope that what I told you today reflects what I learned from Larry, and I am so grateful to him for all those lessons that he imparted that have served me well through every aspect of my career. And I thank him for his complete support and encouragement and confidence throughout all those years, as Leo mentioned, the confidence that he always expressed in me when I was less certain. I wouldn't have said things I've said today back in those days with the same sort of confidence. So I appreciate that, and you know, even after all these years, I feel like I'm still want to live up to his expectations. So I hope you'd approve. And let me also thank, not only was I lucky with mentors, but I've been very lucky with people who I have attracted to my lab, including Leo and many other graduate students and postdocs. I haven't had a chance to talk about the work from all these people today, but especially mention Bob Krieber, my technician, who's been with me for 40 years and still with me. We're both retired. We're still working in the lab on Sunday. And my long-term collaborator, cherished colleague and friend, Chunfeng Wu. We started out as a couple of young kids. When we had this picture taken, we thought we were old. We need the third version. We're really old now, but the two people who most shaped my career were Larry Sandler and Chunfeng Wu. I wouldn't have done the work I did without that synergistic interaction with him. So thank you very much, and I'm sorry if I ran a little bit over, but thank you so much.